A warm good morning to all present here. We have all gathered here for the endowment lecture on current advances in plant disease management in banana program. Now, I request everyone to stand up for the Tamil Thai Valse.
நீராறும் கடலொடுத்த நிலமடந்தை கெளிலொழுகும் சீராறும் வதனமென திகழ் பரத கண்டமிதில் தெக்கணமும் மதிசிறந்த திராவிடனர் திருநாடும் தக்க சிற பிறை நுதலும் தரித்தனரும் திலகமுமே அத்திலக வாசனை போலனை துலகும் இன்பமுற எத்திசையும் புகழ் மணக்க இருந்த பெரும் தமிழனங்கே தமிழனங்கே உன் சீரிழமை திறம் வியந்த செயல் மறந்து வாழ்த்ததுமே வாழ்த்ததுமே வாழ்த்ததுமே.
Good morning to all. Now, I request Dr. M. Theradimani, PhD, Dean, Agriculture College and Research Institute, Kiligalam, to give the welcome address and the introduction of the speaker.
Good morning to one and all. And I welcome all of you for this seminar and presentation of special lectures on management of banana diseases by Dr. R. Thangavelu. I am very glad to introduce Dr. Thangavelu, who is working as Principal Scientist in National Research Center on Banana, Trichy. He has more than 30 years of experience in banana research on, especially specific to fishery wilt of banana, and has more than 12 years postdoctoral experience in banana research. Postdoctoral, that is after PhD, he is doing so many research on banana, more than 12 years. He got awards, so many awards. Jawaharlal Nehru Award, Best Writer Award, High Tech in Banana Cultivation. He got one award, that is a Outstanding Contribution to Science Award, Dr. ABJ Abdul Kalam Scientist Award, Biotechnology Overseas Scientist Award, Outstanding Scientist Award, Eminent Scientist Award for the year 2019 and 20. So far he handled more than 10 external funded projects and 14 in-house projects. So he visited more than 12 countries. He published more than 50 research papers in high impact journals. He involved in the export of banana to Dubai, Italy and UK. He developed a consortium of biocontrol agents for managing the fishery wilt of banana. Now I welcome Dr. Thangavelu to deliver the lectures. Okay. Respected Dean of the Agriculture College and Research Institute, Kiligalam,
and also my dear, my close friend, Dr. Theradimani, the professor of agronomy and organizer of this endowment lecture, Dr. Joseph, and also the professor and head of this agronomy department, Dr. Soleimale, and also the other professors, assistant, associate, and professors of this the great Agriculture College and Research Institute, Kilikulam, and my dear student friends, UG, PG, and PhD. Hope the PhD student also will be there. I don't know. At least the PG students will be there. Good morning to all of you. Thank you very much. Really, it is a very good culture. If any professors or guests or any directors say good morning, nobody will respond in any meeting. At least you have got a very good culture, because how many of you know that I belong to the first batch of BSc Agri in Kilikulam. Super. We belong to Super Killis, 84-88. Of course, we were there, the first year we were there in TNAU for 1.5 years, then we moved to here. Initially, we were at MDT Hindu College. We used to come by bus for 1.5 years or 2 years, and then 1 year we were here. Because of that, I missed some disturbance was there when we were here, and I missed the chance of doing my MSc Agri at Coimbatore. So then I moved to Annamala University for my MSc program, then to PhD at TNAU, then again PDF at Australia, and I did my MBA also in HRA, Human Resource Management. So at the outset, I thank the Dean, AC and Kilikulam, and also the organizers, Dr. Joseph and Solomalai sir, for giving me this great opportunity to express my research experience on banana diseases. So I have traveled widely in different parts of the world. More than 22 countries I have visited. And recently I have been recognized as an expert by United Nations, FAO, International Atomic Energy Agency, Vienna. Recently I visited there as an expert for biocontroller. So let us, without wasting much time, we will go to the subject. I can just slide it. This one? No, this is this one. Okay. Yeah. But pause. Okay. Okay. This one. Okay. No, this one. This one. Okay. But in presentation mode, this is in presentation mode? Presentation. Okay. Okay. Hope that, let me assume that all of you have seen banana. Yes? Eating banana? Very good. That means you need not go to the doctor. If you are regularly eating banana, you need not go to the doctor. Definitely, it is not a joke. Definitely, I am telling, because it contains all the nutrients, balanced nutrients, without fat. But much carbohydrate is there, but the other things are there. Potassium, calcium, vitamins, 10 different vitamins are there. If you take regularly, no problem will come, especially the important diseases like heart disease, cancer, piles problem, stomach problem, so many things it can cure. One banana if you take it. So it is a stable food for more than 400 million people, especially in African countries. and it is a fifth most important fruit crop, and it is grown in more than 120 countries. If we take India, India is the largest producer of banana in the world. We produce more than 33 million tonnes. At world level, the production is around 114 million tonnes. Out of that, 33 million tonnes are from India. The share is more than 26%. But if we take the export, it is negligible. We export only 2 lakh tonnes. So just we started 5 years back the export of banana. We developed a protocol. I am one of the team members in exporting banana. Recently we helped Kerala government to export banana to European countries. Because of my or our technology, I cannot say my because I should not say always my. Because of our technology, using biocontrol agents, if you dip the banana fruit in one of the biocontrol agents, you can store it for more than 75 days. No need of chemicals. So that type of technology we have developed. And you can see everywhere. In India, most of the states are growing banana, except a few states where the temperature is very low. So below 20 degree or 15 degree, if it is there, it is very difficult because in North India, it is a temperate or subtropical regions where the temperature is very low. So there, for 2 to 3 months, there will not be any growth of banana. And the Rajasthan, Kashmir, then Punjab also they grow, but only few, except 4 or 5 states, all states are growing banana. And this is the area of production, area and production of banana in different states of India. And we have got so many wild varieties which are having resistance to so many pest and diseases, particularly in northeastern hill regions, Assam, Meghalaya, Manipur, Sikkim. In all these areas, this type of wild varieties are there. And as far as this share of different banana varieties, how many varieties we are having in the world as well as in India? Any guess from the students? Okay. At world level, we have got more than 6,500 varieties of banana. 6,500 varieties of banana, and in India, we have got NRCB is maintaining more than 360 different varieties. So in Tamil Nadu, we have got more than 20 different varieties. So commercially, what we grow, but the share of different varieties, with regard to share of different varieties, the Cavendish group, Pachapalam, it occupies more than 63% in the case of production. With regard to area, 57% among different varieties. So this is being exported to different countries in large scale. And since at world level also, more than 90% of the exportable banana is the Cavendish. So if anything comes to Cavendish, then the world will be disturbed. Because so many countries, they depend for their economy, depend on this Cavendish for their economy. So stable food, so many things are there. So that is why we give more importance for Cavendish type of banana. If you go to North India, if you travel by train, you will get only Cavendish banana, not other banana. Only in South India you will get different banana, but in Andhra you will get only Cavendish, not other varieties. Earlier they were growing Rasali and others, but now they are not growing. You can see different varieties. If you have not seen so far, you can see how many varieties are there. I think this for you, some provision is to be given for seeing for this screen, no? For the guess, huh? Okay. And you can see different varieties of banana. Yeah. Then we have got ornamentals also. The Musa, Arneta, Gibrina. This now we have given to different parts of our country. Even in Rashtrapati Bhavan, they are growing this ornamental banana, and for receiving any person's bouquet, we can use this ornamental banana. And we produce more than 40 different byproducts out of banana. Pickles, powder, then fiber, so many things. In juice itself, there are so many juices. So biscuit, so many things. Cakes, wine, beer, so many things we are making. More than 44 different products we are making. And fig, this dehydrated banana, this is very good. Even one bunch you can carry in your pocket just by converting into fig. Dehydration. By dehydration, solar dehydration, we can produce fig. So you can become, after completion, you can become an entrepreneur by starting this type of business related to banana byproducts. Then we exported banana to different countries. But what is the problem? Everywhere there will be a constraints. In anything, if you take, even if you grow more than 33 million tonnes and largest producer of the world, but we are facing a lot of problem. This is the crop where you can get more than 3 to 8 lakhs rupees per acre. I am also growing banana, 8 acres of banana for more than 10 years. So last year, the farmers were very happy because they got more than 5 to 8 lakhs rupees per acre. No other crop can give this much profit. I'm telling about the profit. So, but the people are facing a lot of problems because of different diseases, particularly fishery wilt and leaf spot. Leaf spot, you can last week, myself and dean of this college, we went to this area, Kalakad area, where the problem was very severe. We recommended our technology. They have not followed anything, but we have recommended our technology, and we advise the farmers to follow some practice so that they can harvest at least 50% of what they expected. So with regard to constraints, this I was telling about the different constraints are fishery wilt, leaf spot disease, crown rot, anthracnose, erwinia, viral disease. As far as diseases are concerned, these are all the important diseases. This fishery wilt, if you take the fishery wilt, you can see the view of the fishery wilt infected field. And what is fishery wilt? It is caused by a fungus. Everybody knows Fusarium marxisforum, Formis specialis, succubensi. So it present in the soil and in the form of different spores like conidia, microconidia, and then chlamydia spores. Then it germinates and infects the roots, move into the culm and into the pseudostem xylem vessels. Then again, after the death of the plant, it will again come to the soil and survive for a long period. More than 40 to 50 years it will survive. Once if it is introduced, it cannot be eradicated. Only management option, IDM option is only will be working. You can see how to identify. Diagnosis is the 50% of the cure, right? That too early diagnosis. So as far as the diseases are concerned, even for human being or for plant, anything, animals, the early diagnosis is very important. Otherwise, it is very difficult to control or manage the disease. And correct diagnosis. So many pathologists, they don't know even how to identify fishery wilt. I am not joking, really I am telling. So just it is an opportunity for you all people to just learn at least a few diseases of banana so that it will be useful for you. For those who are going to study this PG in pathology will be more useful, and for all professors also, assistant, associate and professors also, it will be more useful because so many other diseases will be like fishery wilt. That's why I am telling. So many wilty diseases are there. So how to diagnose it? The external symptoms and the internal symptoms. The external symptom is just at the margin, you will get the yellowing, slight yellowing. Then it will move towards the margin up to the midrib. Then the leaf will become yellow. The yellowing starts from the lower leaves and moves towards the upper leaf. that is yellowing, then the whole plant will die. This is the first symptom. Then the other important symptom is in the pseudostem you will get the splitting. Even in Erwinia rot you will get splitting, but Erwinia rot very small height, up to 15 centimeter or 10 centimeter only will be there, small splitting. Even normally also, to accommodate the growing sheath and other things, it will split, little bit split will be there. But in the case of fissure wilt, the splitting will be 1 meter height, or it will be more, the splitting distance will be more. And if you cut the pseudostem, and you can see the internal vascular discoloration, that is the xylem vessels become dysfunctional because of brown discoloration, because of production of toxins and enzymes. It has become functionless, the xylem vessels. The function of xylem vessel is to conduct water, right, and nutrients. So once if it is functionless, then the water will not be moved upwards, and because of that the wilting will occur. And internally, if you see, if you cut if you uproot the comb and cut crosswise and see the internal symptoms, you can see this brown discoloration. You can see here, yeah, but it will go up to here. Yeah, you can see here. No. You can see here the internal symptom, vascular discoloration. Here, this region, the round region, is the portion where all xylem vessels join. So once if it is brown or discolored, it will be functionless. And you can see here, this one. So this is the portion where the daughter suckers occur. Daughter sucker, the sucker, side suckers develop from there. So from mother sucker to daughter sucker, how it moves, you see here. So that's why we are recommending that from the infected plant, one should not take the side suckers for planting. Okay, this is vegetatively propagated, no seeds. Seeds will be there in the diploid, not in triploid and tetraploid. So all the most of the cultivated varieties are triploid or tetraploid, but in diploid you will get the seeds, but that cannot be consumed. But some exception is there in Assam, the beam coal, atia coal, they use it for consumption, although seeds are there. So for newborn baby, they will take the flesh of that diploid banana, atia coal, and they will give it instead of mother's milk. To that extent, the banana is being used in different parts of our country. And sometimes mixed infection also will be there. This is the rhizome rot or comb rot disease, and this is the wilt disease. Sometimes mixed infection also can be seen in a single plant. Then how to differentiate fissurium wilt and rhizome rot? So in the case of fissurium wilt, if you pull out the plants, it will not come easily. But in the case of rhizome rot, if you pull out, it will come easily. Okay, the rhizome rot caused by Erwinia pectobacterium. Now they changed the name as a pectobacterium. There are 3 different genera: pectobacterium, Dickia, Klebsiella. Recently we have reported, first report from India, Klebsiella varicola. It causes rhizome rot disease. It has come in plant disease. So you can see here the symptoms. If you cut the common, this one, you can see this type of symptoms. But in the case of rhizome rot, you will get irregular margin. But here regular margin, but here irregular margin. From this region only you can isolate pathogen. Very difficult to isolate Erwinia. I have learned this technique from Australia when I was doing my PDF. So these are all the different, this one, and rhizome rot will come only up to 4 months of planting, after planting. 4 months after planting, up to 4 months you can see this disease. Afterwards, once if it is hardened, you cannot see in most of the cases. But in the case of fissurium wilt, even 1 month after planting, you can see the symptoms, but mostly 4 months after planting. Mostly, in most of the cases, at the time of flowering only, you can see the disease. And pseudostem borer, this will also look like. Why I am putting this slide, showing this slide, because fissure milt and pseudostem borer infected plants look same. The symptoms will be looking same. Because in pseudostem borer infected plant also yellowing will be there, but in the case of fissure milt also yellowing of leaves will be there. How to distinguish? In pseudostem borer, what will be there? From the name itself you can make out. Any guess? Okay. In pseudostem borer, the hole will be there in the pseudostem. Borer means it will make the hole, right? Make the hole. And from that hole, exudation will be there. That is pseudostem. Once if that inner content are eaten by the this grub, nothing will be there. It will be hollow. So no xylem muscles and something. In that case also, yellowing will be there. But yellowing in pseudostem borer will start at any point of pseudostem. Please, this one you have to follow very keenly, because in the case of pseudostem borer, yellowing of leaves will be there, but it will not start from the base. Any portion you can see yellowing, and also holes you can see on the pseudostem. But in the case of fissure milt, no holes will be there. Yellowing starts from the lower leaves, lower portion of the pseudostem. This is the major differentiating characters. And this is the Xanthomonas wilt. We don't have, but it will be there in India also. No expert is available to distinguish this type of diseases from fissure milt or other things. That is a very pathetic situation in India. So the bacterial wilt it is caused by three different genera. One is the Pseudomonas. Its name is changed as Ralstonia solanacearum. Then second one is Xanthomonas, and third one is Erwinia, the Pectobacterium. There are three different genera causing bacterial wilt disease, and this is one of the wilt disease, Xanthomonas wilt. This is present mostly in African countries, Xanthomonas wilt. What is the major difference between fungal wilt and bacterial wilt? Anybody can tell? Without? Very good. See, when I was taking the class in IRA recently 2 months back, no one could be able to tell. Very good. Thank you for your. I was asking. Very good. So oozing, bacteria means oozing will be there. If you cut the pseudostem, even the professors, I was taking class for professor craft training, as all professors from different parts of the state, including Annamalai University and TNAU, but no one could be able to tell. So at least you are studying PG, right? MSc, first year, pathology, plant med, very good, very good. Okay. So here the important symptoms are wilting. Wilting, in the case of wilting, yellowing will be there. Yellowing of leaves will be there. Then irregular ripening. Just bacteria, I am distinguishing the symptoms of bacteria from fissure milt. In the case of fissure milt, no irregular ripening of fruits. But in the case of bacterial wilt, irregular ripening will be there, important symptoms. The second one is the oozing. If you cut any portion of this one, petiole or pseudostem, the yellowish brown oozing will be there. But in the case of fissurium, no oozing out will be there. Then third important point is the suckers will be rotten in the case of bacterial wilt. But in the case of fissurium wilt, the suckers will not express any symptoms unless you cut the suckers and see the internal symptoms. The external symptoms will not be there in the case of fissurium wilt in the suckers. But in the case of bacterial wilt, you can see the external symptoms in the suckers. This is very, very important characters, distinguishing characters. Then another disease, this first time in the world I have reported this occurrence of this disease called, this also like initially I named it as a Trichy wilt because I identified in Trichy. Then I changed it to Comrade disease caused by Sclerotium ralphsi. This is spreading very fast in different parts of the country, and Even repeatedly, if I tell that it is caused by sclerosis, people are not remembering it. So the very simple sclerosis neuropathy, the sclerosia-like structures, you can see it in the affected portion. The mycelium will be there and sclerosia will be there. Very easy to distinguish. So this is caused by, you can see how the calm has been converted like coconut. If you use even your arwal which vetna kuda, you cannot, to that extent it will be modified to fibrous, the calm. So you can see here mycellar growth, then yellowing. So for any ordinary person or any pathologist who have not studied this, worked on this banana wilt, they will say that it is due to fishery milt. But this is not due to fishery milt. You can see the mycellar growth in the calm as here, you can see here. Okay. These are all how to, so far what I have told is how to distinguish the fishery milt from other wilt diseases which also cause similar symptoms like fishery milt. Okay, now are you clear? Are you clear? Huh? So, okay. If you have any doubts, you can ask afterwards. Okay. Now, with regard to fishery milt, there are 3 different races. That's what your professor would have taught you. There are 3 different races. Even Dr. Ebenezer sir is also there. Race 1, race 2 and 3 and 4. 3 will not cause disease in heliconia, but race 1, 2 and 4 will cause disease in banana. But that is not the actual thing. Because of my finding in 2010, okay, before that I want to tell you how races are differentiated. Based on the differential host, the host will differentiate the races, this one, the races, different races. So if a particular Fusarium strains infect particular host, then it is called race 1. In the case of race 1, it will, you might have studied that race 1 infects Carprovoli, Rasali, Grove Mitchell, all these. And race 2 infects cooking banana. And race 4, it infects everything, including Cavendish banana. That is the thing what we have learned. But because of our finding, it is not that one. Race 1, race 2, race 4 classification is not at all there. It is only vegetative compatibility grouping. Because the race 1 is also infecting Cavendish. It's supposed not to infect Cavendish, but because of from our finding, this race 1 can also infect Cavendish. First time we have reported from Thane area, the Cavendish, what the fishery milt, what they are getting in Cavendish is due to race 1, not race 4. So the race 2, cooking banana, and race 4, everything. It infects all the varieties. It will not distinguish. So this is the race classification, but what you should know is race 1 also can infect Cavendish variety. You can see the gun what I have put is virulence to denote virulence. The race 1, it is compared to other TR4, tropical race 4 or race 4, it is less virulent, race 2 also. And what I have identified, this has got more virulence compared to race 1, ordinary race 1 and 2. 3 number of guns denotes more virulence. Okay, more number of guns, more virulence. Okay. So this infected Cavendish and tropical race 4, this has got so many guns to affect variety, different varieties. So this tropical race 4, this is very important throughout the world. It has spread to more than 21 countries. And in India, I have reported this occurrence in 2015. I have identified, but I was not allowed to this one to publish it because of the sensitivity of the problem. I was allowed to publish only in 2019. So I reported this occurrence of TR4 in India in 2019, and so far it is there in 21 different countries and causing huge loss. If anybody interested to take pathology as a subject in PG, you should work on fishery milt. You have got a lot of scope to work in abroad. And you can see the map. You can see the blue color, it is race 1, and this yellow color is subtropical race 4. In race 4, there are two categories, subtropical race 4 and tropical race 4. Tropical race 4 will occur in tropical climate condition, and subtropical race 4 is that requires cool temperature to infect the crop. Subtropical region only it will be there, subtropical race 4. But in our case, the tropical race 4 is there in subtropical regions, the Bihar and Uttar Pradesh. That means why it is there, that I will tell you afterwards. But now you just see the distribution of the different races. Tropical race 4, subtropical race 4 and race 1. Tamil Nadu, Karnataka, then Maharashtra and Gujarat and Madhya Pradesh. In all banana growing areas, race 1 is infecting Cavendish. But the red color, it is tropical race 4 infecting Cavendish. It is identified in Bihar, UP and West Bengal. In these 3 states, we have reported tropical race 4. You can see the devastation done by the tropical race 4 in Bihar and UP. You can see there. Yeah. So I was telling you about the disease cycle. The spores present in the soil infect the roots and move to corm, then phloem, then again come to the soil and infect. How it spreads? Through water, through bunch, that is through peduncle. Up to the peduncle, crown portion it will be there. The fungus will be there up to the peduncle region. So if you transport the banana as bunch, then it will spread to different areas. Then soil adhering to the tires and others implements. Even if you walk through the infected field, if soil is adhering to your shoes and chappals, then you may carry the spores and spread to the other uninfected areas. Even this one, all implements, whatever implements you use, even if you pull out the plants and put it in the irrigation channel, it will spread. Even in the food, you can see he is carrying some amount of soil that may carry the fungus also. And tires, even animals also carry the fungus, spread the fusarium. And infected suckers. In most part of our country, they use the suckers for planting as a planting material. So they are spreading very fast. Even tisculture also, if you set up tisculture unit in the hotspot area of fusarium, that may also carry, that will be contaminated. Tisculture plants also will be contaminated. The soil what they use, the water what they use for hardening, for growing the tisculture plants, that may contain fusarium. And it is, nobody knows, so far nobody not knows, they may not be knowing that it is airborne. So recently they from abroad they identified that it can be airborne also. So what is the aggravating factor? What are the aggravating factors for fusarium wilt? If you cut the whole plants after harvest and put it in the field, that may aggravate. And this stem borer also carry the fusarium in the exoskeleton. And nematodes. When nematodes are more, the aggravation will be more, severity will be more, because it causes some wounds in the roots and corm. So through wounds also it will move, through the natural opening as well as wounds created by the by human being when they are doing intercultural operation, or by the nematodes or weevils. It may enter through the wounds, it may enter. So the loss, what would be the loss? The worth of banana what we grow in India is 50,000 crores. But because of fusarium wilt, even if you take 20% as incidence, we are losing 5,000 crores per year. Even this will be more in northern India. Just imagine, 5,000 crores means what? So it is a loss to the farmer. And how to characterize? Suppose if you are seeing some plants infected with the fusarium wilt, how will you characterize it? You have to do pathogenicity, then This is the pathogenicity you have to grow and inoculate and see Cox postulate you have to prove. If it is infected, then it is a pathogenic to this plant. Then vegetative compatibility grouping. I don't want to go in deep because you are not the correct audience for me to explain everything in deep. If you are a pathologist of MSc or PhD, then it would be of great importance. But now, just so you know what all the techniques are there. Because in India, in the world, only 4 research institutes are carrying out the characterization of Fusarium wilt. One is the ICAR NRCB, then second one is Australia, third one is South Africa, and fourth one is USA. In these 4 different places only, this characterization is going on, and by using the marker, So these are all the different techniques, and sequencing also. By sequencing elongation factor alpha 1 gene for Fusarium, this is specific to Fusarium. If you isolate, extract this one and sequence it and going for phylogenetic analysis, then you can identify. So this is one of the methods. Yeah, just volatiles, simple technique, just to grow it in rice medium and smell it. If you get odor smell, it is race 4, and if you are not getting any odor, then it is race 1. By that way also you can characterize, but it is a crude method. Yeah. Then we have done some sequencing. Just let me go fast because our Fusarium is distinct from others. What is available in abroad? Our this genome size is large in our Fusarium, and we have identified the variations in chromosome 3, 6, and 14, 15, where the SNPs and indels were more. Because of that, these chromosomes are lineage specific chromosome for Fusarium, where you can get all the virulent genes in these chromosomes. So then these are the different protein coding genes what we have identified through whole genome sequencing. Then you can see here the Venn diagram. You can see the virulent genes are effector genes associated with tropical race 4, race 1, and subtropical race 4. The unique genes and common genes. You can see here. Another one is effector genes. Effector genes, it is called SIX genes, secreted in xylem vessels. The effector genes is very important for pathogenicity, and it is more specific. Why we have done is, our Fusarium is different from others. Everybody says that it has spread from other foreign countries. It is not. It has evolved here itself. So by analyzing the SIX genes, since it is very specific to the particular strain, we have designed the primer from these effector genes. Whatever is available, it was designed from the ITS and IGS region, which is not more specific to particular strain. But this SIX genes is very specific. You can see here SIX7 is very specific to subtropical race 4. And for tropical race 4, SIX8 is more specific. So from this, we have designed our own molecular marker. And this is, I was telling to you, just one point I want to stress here. There is a variation here in SIX gene. The SIX1 gene present in chromosome 14 has got variation in amino acid sequence by deletion. If any deletion, if you notice in amino acid sequence, then it is more virulent. That's what the reference says. So why our race 1, which supposed not to infect cavities, why it is infecting here? Because of this point, you can see here, the SIX1 gene, it has got polymorphism in amino acid position of this one. What is this? 205 to 214. So if any deletion is there in that, it will become more virulent. That's what our finding says. And from this, whatever the marker available in abroad, it was not working for us. This also we have identified and reported. So we wanted to develop our own molecular markers, and we designed our molecular marker, and it is working very good. So by using our molecular marker, simply you can identify the fusarium, what it is. Then management. Different management practices are there. The quarantine and sanitation. Quarantine stations are there, namesake, everywhere. You might have studied quarantine, but it is not at all. No one bothers. No scientists who are working in quarantine station, they don't bother for controlling the spread of the disease. So we have to, first the important point is we have to create awareness. That's what we did, myself and your dean and our director. We did the awareness creation in Kalakad area, what it is, how to control all these things. We have to create awareness, the loss expected due to this disease. All these things we have to explain to the farmer. Then quarantine stations we have to create. Then we have to immediately kill the infected plants and destroy it just by injecting Roundup. We decide. Then decontamination of all implements what we have used. Then keeping the signboard. We have to keep the signboard, but in our democratic country it will not work. But some progressive farmers are there. I have got this disease, don't come inside. That type of, because once if it is introduced, it will be there forever. How to control it? But we have got control measures, biocontrol measures. You can see how they are. We only taught. I traveled all the areas of banana growing states in India, and we conducted large number of lot of meetings, everything, and created awareness. They are injecting and they are destroying. How they are destroying? Just putting the rice case and the waste of the banana. You have to destroy the infected plant. Then cultural control, flood following. Then disease free. We have to take the suckers from the disease free plants. Then crop rotation. This is very, very important. If you go for paddy for 1 year and then go for banana, even if field is infected, in the first year there will not be much symptom, much incidence. Second year it will build up. Then adaptation of drip irrigation, then soil amendments, then chemical control. The carbon dioxide is working very good. And then the biological control. This is very, very important. Simply see, what you should know is whatever the trichoderma available in the market may not be effective for a particular disease unless it is tested against a particular disease. Just please understand this only one point. Whatever the trichoderma, pseudomonas, any biocontrol agents available in the market, unless it is tested against a particular disease in hotspot area, it will not be working. Don't recommend. So we have to isolate. We have isolated 300, 400 of different trichoderma, pseudomonas, bacillus, so many things. But we have come out with only 1 or 2 trichoderma isolates. How? By screening for different mode of actions, spore germination, mycelial growth inhibition, then citrophore production, protease, chitinase, lytic enzymes production, everything. So whatever we isolate, that should have multiple actions as well as multiple functions. That means it should not only control the disease, but also it should promote the growth of the plant. That type of versatility of the biocontrol agents we have to identify. So this is how we have to go about the screening and all. So you can see out of 170, this is gone. Hold on. Huh? Okay. For growth promotion, IAA, phosphate solubilization, zinc solubilization, all these things we have to do screening for all these. Then out of 176, and another important thing is just simply you cannot take the soil and then isolate and then apply biocontrol agents. Because some organisms without microbes, no one can survive in the world, even human being also, without microbes. We have got a lot of microbes in our gut, everything. That is only helping us to survive. Even for plants also, the beneficial microbes which are present in the soil. It is helping the plants to survive. It is protecting the plants from pathogens. So we have to, the term is called soil health. The central government has introduced a soil health card without biological properties. That is a failure. They do only physical and chemical properties of the soil, not biological properties. Biological properties only is very important. So out of 176, only 11, sorry, I wanted to tell you endophytic. Endophytic is very, because it enters into the system and survive there and protect the plant. Like in our house, watchman alone will not be sufficient. Somebody also should be there inside the house. If they enter inside, how watchman will protect? So inside as well as outside, rhizospheric as well as endophytic organisms we have to isolate, and then we have to deploy. So by doing all these things, endophytic, bacterial, fungal, and then rhizospheric, bacterial and fungal, all these we isolated, screened for different mode of actions, for different growth promotion activities, and we have come out with these two consortia and also the botanicals, GIMO. GIMO, it is a cross between onion and garlic. So in China, they are using it as a Chinese leek as vegetables. Even if you use this GIMO as vegetables, you will not get heart disease because it contains gallic acid. So this combination, it is working very good for both race 1 infecting cavities as well as a tropical race 4. For any fishery wilt, these two combinations are working very good. And how we have multiplied in rice chaffy grain, in 5 days we can mass produce. Even if you isolate very effective biocontrol agents and if you sell it for 200 rupees per kg, no one will purchase. The farmers are very poor. They will hesitate to purchase. If you develop some technique using the cheaply available material in the farm holding itself, and if they themselves multiply and apply in the field, that would be very good, great. That technology will spread faster. These rice chaffy grains will be available everywhere. They are simply throwing it. After winnowing, you will get rice chaffy grains. In that you can mass produce. And you can apply. This is GIMO plant. Just like onion. You can cut and put it and then it will grow like that. As intercrop, if you grow, it will reduce inoculum to a higher level. So this is called bioframing. The whatever you multiplied the trichoderma, you put it before planting. You put it in the tisculture plants. Allow it for 5 days or 1 week so that it will enter inside and occupy the all rhizosphere region. Then you plant it, then it will protect. If I am saying like this, don't think that it is very simple to control the fishery wilt. Okay? Very difficult. The world is struggling. More than 5,000 scientists are working on this. They are spending crores and crores of rupees. Recently, I got 1.25 crore for the management of fishery wilt disease as a project from United Nations. On 17th, I am recruiting one PhD fellow for this purpose. So this field is abandoned by the farmer in TNAU because of fishery wilt. In TNAU, most of the farmers, they abandoned the field. In 2010, I identified this field. I wanted this field and I developed biocontrol agents, applied there, and botanical also. You can see how this was converted. You can see the abandoned field to full-fledged, the field with full of banana plants without incidents. You can see there. And not only that, the size of the bunches because of growth promotion, 45 kg. But in the case of control, only few plants have given bunches, and that too with 5 kg. You just imagine the effect of application of biocontrol agents, endophytic as well as rhizospheric. Then mass production, different methods we tried. Initially using rice chaffy grain, then FIM, then very cheaper method we have developed recently. By just spending 5 rupees to 10 rupees, you can mass produce your biocontrol agents and apply for 5 acres. I'm not joking. It is for 5 acres. Just imagine 10 rupees to 5 acres. Otherwise, the farmer has to spend 80,000 rupees to 1 lakh rupees for applying to 5 acres of land. And also, this shelf life, if you just keep it, this is a liquid formation, and if you keep it for 8 to 10 months, it will be the CFU will maintain as such. Now, another important thing is, by this technology, the fermenter for mass production of bacteria is not at all required. Fermenter, which is costing 5 lakhs to 50 lakhs or 1 crore, that type of equipment is not at all required. Just by spending 5 to 10 rupees, you can mass produce both fungus and bacteria. And how we have demonstrated in Bihar, the growth, and you can see the shelf life, the CFU after 8 months. Just so you can keep it. Even through drip irrigation, you can apply, because labor availability is very difficult nowadays. You have to give a lot of amount as well as 2 vada and 1 coffee. Without that, even if they will forego salary and not the vada and tea. I don't know whether in Kilikulam this is there or not, but in Madurai and Trichy, this is this type of practice is there. So without bothering for labor, just you can simply put it in fertigation tank and you can apply through irrigation. So we isolated principal compound. The what compound it is involved, we isolated through different methods, and we identify. You can see 5 different methods from botanical as well as trichoderma. That is the compound. What you are getting position and other things like that is the same compound, which is effective against specimen to be isolated, and you can see the effectiveness under pot culture as well as in vitro condition. Yeah. So, but further we couldn't, because nanotechnology also we tried, because it is costlier. The principal compound what we isolated is costlier. It is very difficult to apply. We have tested under pot culture condition. It was effective, but for field condition, 1 gram 3,000 rupees means who will purchase? Very difficult. So when I went to USA also, I asked so many scientists in Boston for this phytopathology seminar. I went there. I asked the scientists. No one could able to give answer for this. How to apply this principal compound isolated cheaply. Then the host resistance is also very important for keeping the plants without breaking the resistance. Biological control is very important. If you grow resistant variety continuously for 3 to 4 years, it will break the resistance. The pathogen will break the resistance, it will become susceptible. Unless you follow IDM technology. IDM, integrated disease management technology, is very, very important. In that, soil health is very important. In soil health, use of biocontrol agents which has got multiple actions and functions are very important. This is the take-home message for you people. So host station we have screened under hotspot condition in both in TNAU and Bihar for race 1, race 4, and we have come out with 50 to 60 resistant plant, but all cannot be grown. Some can be grown, some can be used as a resistant source. This has been published in Nature, one of the Nature journals, scientific reports, which has got a high impact factor. So this is Sugandham, very good flavor. You should taste it. So many, we have got so many bananas. Sugandham, we have named it as a Sugandham. In Kulli Hills, they are growing in Kulli Hills, and it was devastated, completely extinguished. And we developed methods for the management and regenerated. Now they are growing happily. The Sugandham. The NCR 17, Nendran, you might have tasted Nendran. It is like both Nendran and Pachapalam, Cavendish. You can eat it, not like Nendran, but like Cavendish, you can eat it, this NCR 17. Then through mutation breeding also you can develop varieties. By subjecting the explant, shoot or embryogenic cell suspension culture for this chemical as well as physical mutagens, gamma radiation, then EMS, ethyl methane sulfonate, methyl methane sulfonate, N-methyl, N-nitroguanidine. These are all the chemical and physical mutagens. By that way also we have developed some varieties. And you can see Rasali we have developed, and also in Cavendish we have developed a resistant variety against Fissure mildew disease. And in abroad also they have developed some varieties GCTCV, but they are claiming that it is resistant, but it is not resistant. It is a moderate resistant. It is expressing 20 to 30% incidence. GCTCV, this is being grown widely in different countries of the world. And recently from Australia, they have developed one transgenic plants by taking the gene from resistant RGA, resistant gene analog 2, that was taken from the diploid. I told you the diploid can be used as a resistant source. Musa acuminata subspecies malaxensis, Calcutta 4, that is called Calcutta 4. From that they taken the gene and put it in the Cavendish, and it was showing resistance. And this was from China. When I went there, they have shown this ZJ 9 and 4. This was resistant to tropical trace 4. And this is the different approach, how to improve the soil health. Crop rotation you have to follow, definitely intercropping, cover crop, mixed cropping, waste recycling. I want to just tell one point. If you apply 50 bags of your potash, particularly potash, 25 bags will be available in the plant. After harvest, the remaining plants will be there. In that, 50% of what you applied will be there. That you should recycle it and then again put it in the banana field, so that you can reduce the fertilizer application to 50%. This is very important. And then effective microbes, soil amendments, all these things you can follow. I think for Fissure mildew I have taken this much. Quickly I will. Leaf spot disease. This is it is airborne, caused by Mycosphaerella. And again, here again, just another, how much time I took? 11:30. Okay, this is very, very important. Because last time you went there for this. For any plant, for banana plant to get optimum yield, please listen. Anybody is having this one, sleepiness? No? We can clap. Yes. Okay. Over. Once again. No? Okay. Thank you. Okay. Really, from the bottom of my heart, I am telling you are a very good student. You people are a very good. Really. And all the credit goes to whom? Dean of this college. Thank you very much. Okay. Now at least for another 10 minutes you will not get any sleep, right? I was when we both were working as SRF, I was taking class for MSc and PhD. MSc. So during our period only we have introduced transparency, overhead projector for teaching the student. I was a little bit strict. I was very strict at that time. But thing is, I will be strict, but I will put marks. Still I am following. What do you want? I will ask. 29, 30, but they will not be knowing. After seeing the mark sheet only, they will come to know. 29, 30. They thought that only 18 or 20. Still, we have to write the annual assessment report. In that, very good, excellent, all these things will be there. Always I put the extreme end. I will ask from them, but at the same time, I will expect that they have to work work hard. Okay. Okay, with regard to leaf spot disease, the companies to sell their product, they were cheating the farmers, particularly in Maharashtra, where the banana is grown in more than 50,000 hectares, 1 lakh hectares. Their life is banana only. They are simply cheating that Mycosphaera fijiensis, black Secotaka, it has come. Secotaka, have you heard the name Secotaka? Huh? How many of you have heard the Secotaka leaf spot? Raise your hand. Second year, okay. Now, sir, because sir is also there, at least it is a very fortunate thing that sir is also there, because he will teach afterwards now for you that in India, Secotaka leaf spot is not there. It is only Umusae leaf spot disease. Umusae means Mycosphaera umusae, not Mycosphaera mesicola or Mycosphaera fijiensis. No, we don't have. For the past 50 to 70 years, people were taught like this only. We have got, I was taught like that, Secotaka leaf spot disease. In this same building here, not this building, next building. There were only 4 buildings, right? So I was like a mad to study pathology. So many things are there. Okay. Okay. Leaf spot disease, for getting optimum yield during vegetative phase, there are 3 different phases: vegetative, flowering, and maturation. During vegetative phase, there should be at least 15 to 20 green leaves without leaf spot for getting good yield, normal yield. Whatever this fertilizer you will give, it will not be useful unless you maintain green leaves. During shooting, 10 to 15 leaves should be there. During harvest period, minimum 10 leaves should be there. If you maintain this, you will get good yield. But because of leaf spot disease, only 2 to 3 leaves will be there during shooting or during harvest period. Just imagine. It not only reduces the yield, but also it causes reduction in quality, size of the fingers, early ripening. It will lead to another disease called peduncle rot. Because peduncle rot, it is caused by sun injury. When injury is there in the peduncle, injury is there due to sun, the pathogen, Polytrichum and Glaciodiploida will enter inside and cause rotting. And bunch will not develop. Once rot comes, then bunch will not develop. So the leaf spot disease not only reduces the yield and quality and early ripening, but also lead to another disease called peduncle rot. So we have to control this disease. You can see there, it is burnt like appearance. How see, it was agitated in Parliament. You will not believe, in 2009, 10, 11, all the MPs from Maharashtra and Madhya Pradesh, they were agitating in Parliament. Immediately, the then Union Agriculture Minister, Mr. Sarat Pawarji, how many of you heard his name? He called me. He called me to his chamber for 3 times, and he has not allowed any DDG or director, anybody. He allowed me only. He discussed for 3 times, and he took me to Jalgaon, Maharashtra, and he introduced my technology. The technology which I am going to tell to you, that was introduced by him in Jalgaon in a big gathering. More than 2,000 farmers were gathered. And once he translated, he asked me to tell in English, and he translated into Marathi, and he explained to the banana farmers in Delhi. To that extent, he was very keen to control the disease and help the farmers. The Union Agriculture Minister Sarat Pawarji. You can see there, you can see the severity of leaf spot disease. Early ripening. Once if it ripened, no trader will take your banana bunch. Even few fingers ripened, very difficult to sell it. The peduncle rot, you can see there, particularly in Caprol and other, this one, it will come. So how to distinguish? Very difficult. Mycosphaera, nowhere in India this culture is available. I can challenge even in IITC, IRA, except in our center. We only know how to isolate. Nowhere this can be isolated. I used to supply the cultures to different parts of the country for doing research. So Mycosphaerella, there are 3 types, Eumuse, Musicola, Fijiensis, and we have got only Eumuse species. You can see the different morphology and color of the culture, and the spores you can see there, and how to distinguish. Just after isolation only you can identify. How to identify? This germination, polar germination and lateral germination. But here in Eumuse, both polar as well as polar means on both the ends of the spore it will germinate, and lateral means side. So in Eumuse, both polar and this one lateral, and we have developed molecular markers, very effective molecular markers, and by that way, by this technique, just simply take the leaf spot, grind it, take the DNA and put it for molecular identification. That will work. That type of technique we have identified, that the LAMP technique, loop mediated isothermal amplification. Just simply within 2 hours you can identify what it is. Just by seeing the color. The green color, fluorescent color indicates that it is positive. That is Eumuse. The other one, it is not there. This is the steps involved. Just heating it in 65 degrees centigrade for 1 hour. That's all. You will get the result. How it spreads? Because it produces ascospores as well as conidia. Through air it will spread, that is windborne, and also through infected material. So how to control? Cut the leaves periodically and destroy it. Then wider spacing, optimal spacing. If any water stagnation is there, provide drainage and go for this spray schedule program using mineral oil and fungicide. This was introduced by our minister in Maharashtra, and if you follow strictly, you will not get any disease. Definitely you can expect the number of leaves what I have told to be maintained. So this is this one, and it is very cheap also. Why we have to use mineral oil? For banana, for any spray you have to mix sticking agent, spreader or adjuvant you have to use, otherwise it is not effective. But in the case of mineral oil, if you use mineral oil, no need to add sticking agent or adjuvant. And it has got fungistatic effect. Even if you use mineral oil alone, it is effective. Because in organic farming, this is very much useful. Then it is compatible with all fungicides, and it will not be washed off by rain or something like that. So this has got this many advantages. And we have isolated our endophytes, microbes. This is being used by KVK Karur, and this is also very effective. And Jimmu, Sarvaroga Nivarani, Jimmu and Trichoderma. So these two for banana, even for other crops also you can use. Jimmu, if you grind it and apply it, it will control fusarium, it will control leaf spot, it will control Cultotrichum, it will increase the shelf life, it will control nematodes. What else is required? So that type of things we identified. And freckle leaf spot, this will come in cooler region. In Hosur and other areas, once if this comes, that's all. They cannot sell it. To that extent it will cause. And any exportable banana, any banana which is exported contains this type of freckle spot, it will dump in the sea only. They will not allow to enter inside. So freckle, without freckle, it should be exported. Banana should be exported. So this is also very important disease, and this is also very difficult to control. Nobody knows the methodology to isolate even leaf spot also they can know the technique, but they cannot isolate. But freckle, no one knows the technique to isolate. It is a specialized technique, and you can see like sandpaper. Sandpaper, if you rub it in your hand, that feeling will be there in the banana which is infected by freckle. You see there, Yeah, that how many of you have tasted hill banana, Virupakshi, Sirumalai? Except the Dean, because he is from that place, Tindikal district. Any other, any, no one has tasted Sirumalai, Virupakshi? Very costly. Panjamirdam have you taken? Panjamirdam. Huh? Can you raise your hand? How many of you have tested Panjamirdam? Okay, very good. Hope all might have gone to Palani and got Panjamirdam at least. Okay, at least Tiruchendur you might have visited, got Panjamirdam. It is mostly from that banana only, Virupakshi. So that is always, it is black color, not at all a green color. It is that black color is due to freckly spot. So management, whatever the Bevision and other things, whatever you use for management of leaf spot, that will take care of this one. Then cigar and rat, copper fungicides, if you spray, it would be good. You can see the cigar and rat, like a cigarette. At the end, you will see the ash type of things at the end of the cigarette. From this side, might have seen. So that end, the same appearance will be there in the banana fruit which is infected with the cigar and rat. Surut. Yeah. Very familiar with the surut. Peduncle rat. Peduncle rat. This I told now, when more number of green leaves are not there, the sunlight will directly fall on that peduncle and it will cause injuries. Like if then we will apply something. If you don't apply, then we'll get fever and other things, because some bacteria or fungus enters inside the wound and creating all the troubles. Same thing it will create. So peduncle rot, once injury is there, the coltotrichum, which is there as a latent infection, coltotrichum and raciotyplodia will enter inside and cause rotting of the peduncle. So bunch maturation will not be there. This is the anthracnose. If you eat the anthracnose infected banana, we will not get cancer, particularly the breast cancer. It is already published from UK. If you take this type of anthracnose infected banana, because the anthracnose infection in banana will lead to a production of a compound which will inhibit or prevent the occurrence of cancer. Yeah. So for this, for control of anthracnose, we will use Bevisin everything. We will dip the banana in Bevisin, which is carcinogenic. So for that, I have used the same Trichoderma aspergillum, which is effective against Fusarium wilt, for the control of anthracnose. It is working very good and it increases shelf life up to 75 days. So chemical-free banana can be exported by our technology. Even Jimmu also. This is a bacterial rat. So many things are there. How to complete? You can just see the Erwinia rot, affected tree, and the symptoms are then management, just bleaching powder. You apply bleaching powder, then you apply all other soil amendments and bioconvergence, whatever is there, that will take care of. I cannot explain everything because of lack of time. This is the bacterial wilt I was telling to you. Just important symptoms I will tell you: yellowing of leaves, irregular ripening, then bacterial ooze. You can see everywhere. And it is transmitted through insects. It will enter through flower as well as from the bottom. Through the roots it will enter, the bacteria. The blood disease, this is called blood disease. If you cut it, people even they will think that, oh, God has given this one and blood is coming. All these things, so many things has happened. So by cutting, blood-like things will ooze out. So this is caused by the Ralstonia Syzygy celibensis. This is present mostly in the Indonesia, Malaysia, and other South Asian countries. But Xanthomonas wilt is more problem in African countries. And this is sclerosis I already told. Then viral disease, you know very well. Just this alone. This is a banana bunch top. You can see the symptoms here. கொத்து முடி கொத்து நோய் மேலே டாப்பில் வந்துட்டு just it will be there. When you are going for this third year and final year, at that time you will be remembering me. Oh, that the day he came and showed all this. So you will get a full mark in banana diseases. I hope you will get full mark. This banana budget of disease, then bracts mosaic virus. In the bract, you can see the mosaic pattern. This is CMV and this is banana streak virus. For all these things, what you have to do is, don't take suckers from the infected one. Apply more amount of fertilizer, organic and inorganic fertilizers. That will, and for insects, you spray imidacloprid or some monocotopas. That will manage the viral diseases. This is a multiple actions I was telling to you. It controls whatever the biocontrol agents I identified, the trichoderma or bacillus. It controls nematodes also. It controls post-harvest disease. It controls yeast spot. It controls fishery milt. You can see that's what our scientists have tested. The Trichoderma aspergillum can also manage the nematodes like root lesion nematode and root knot nematode. This is what required from you. If you are studying for pathology or something like that, you have to look for this type of approach. All in one. Okay, that type of approach, if you have got, if you develop, it would be a very good advantage for not only for the farmers, for the country. Thank you very much for your patient listening.
Thank you, sir, for sharing a lot of information regarding banana cultivation and various diseases in banana. Now, we request Dr. M. Theradi Mani, Dean, Agriculture College and Research Institute, Kiligalam, to honor Dr. R. Thangavelu, Principal Scientist, ICAR, NRCB Trichy, with the momentum. Thank you, sir. We request Dr. A. Solemalai, PhD, Professor and Head, Department of Agronomy, Agriculture College and Research Institute, Kiligalam, to honor Dr. R. Theradi Mani, Dean, Agriculture College and Research Institute, Kiligalam. Thank you, sir. Now I request Dr. A. Solemalai, Professor and Head, Department of Agronomy, to deliver the votes of thanks.
Respected and honorable
Any questions from the audience?
If you have got questions, you please ask me, but minimum 10 questions. Minimum 10 questions. From this side 5 and this side 5. Equal. Even if you express that, I have not understood this. Lecture also.
So first of all, I express my heartfelt thanks to providing this type of excellent opportunity to hear your experiences in banana management, disease management, sir. I have some few questions, sir. During your presentations, you are told that anthracnose infected banana will help to prevent cancer.
Yeah, anthracnose infected.
Yes, sir.
Yeah.
Then why we have to control this anthracnose?
Yeah.
Because it's providing some medicinal utility.
Yeah. Right. Okay.
Nowadays, cancer is a big problem in human health consideration. Then
Yeah. Okay. But at the same time, the anthracnose infection, if it is there, then it will spoil the entire fruit. The stages are there. Suppose there are 6 different stages. At third stage, if you eat, the glucose level will be lower, and in the fourth and fifth, the anthracnose will develop, and in sixth stage, it will completely spoil. So, because now only the awareness is created, but the other than that, the crown rot and other diseases are there. And the European people, mostly the bananas are exported to European countries and also to Japan and China. They like only disease-free food. But how many of you they like the anthracnose infected? Because the awareness is not created so far. And for the time being, we are maintaining the disease-free food. And if you like to eat this one, it is good. That's what we want to tell. Don't discard it. And if you got anthracnose infected banana, you please take it. That would be good for your health, and it would not have any disadvantage or harmful to your health. It would not cause.
Thank you, sir. And another question is, so you have told that
This cannot be included in 5 questions because he is from staff, right?
Yes, sir.
You are from staff side. Okay.
So you have told that fusarium spores are spreading through adhering soils with tractor wheel or shoes like that, sir. How long it will be persistent, sir? What is the survival?
Okay. See, this tropical race spore was identified in 1990. From that years onwards, people are counting. So far, 10, 20, 30, 33 years are over. Still it is surviving. We don't know how long it will survive. After 30 years, even if it survives, then the data will show that up to 60 years it will survive like that. So if you do management practices, the inoculum level will be reduced. You cannot completely eradicate it. It will survive. The chlamydia spores, what it produces, it survives in the soil. And if a host is not there, it will survive in weeds, which will not, the weeds will not express the symptoms. Non-symptomatic host. It will survive in non-symptomatic weed host for a long time.
Then you have shown many banana varieties, sir. Which variety is more resistant to fusarium wilting, sir?
Which variety?
Which variety of banana is more resistant to fusarium wilt, sir?
The commercial variety.
Commercial variety.
Nendran and red banana. Nendran and red banana. These two are so far showing resistance. I cannot say that it will be resistant. Showing resistance till now to fusarium. Red banana and Nendran, that's what the TNAU farmers are practicing it. Of growing Nendran and red banana in wherever the fusarium problem is there.
Another common question, sir. So in the market we are having different bananas, sir, like Morris and Cavendish. These Morris and Cavendish are same, sir, or different?
It is same. It is a Cavendish type. There are 3 different types: dwarf Cavendish, Cavendish, and giant Cavendish. This Morris comes under dwarf Cavendish, and our what we eat is coming under Cavendish, and giant Cavendish, the Robusta and all, it is Cavendish. Then the giant Cavendish is the fruit, the Cavendish type, Grand 9. That is called Grand 9. In Bangalore and all, it is available. But earlier we were growing Robusta, but nowadays no one is growing Robusta. Cavendish is, giant Cavendish only they are growing. So there are 3 different types.
Okay, sir. Thank you, sir. Thank you, sir.
Good. Thank you very much for your question.
Thank you for an exhaustive and informative lecture. Sir, you have mentioned about the Simmo.
Jimmo.
Yes. It is botanically Allium ruberosum.
What you are?
I am Rosen Head, Department of Horticulture.
Very good, very good. Okay. You both plant the same.
You have mentioned about Simmo, sir.
Jimmo.
It is actually botanically Allium ruberosum. It is a rich source of sulfur compounds. Allicin
Yeah,
and sulfur compounds.
correct.
how it controls diseases. You have mentioned it's a remedy
Yeah.
for most of the diseases.
No, the most
and how
of-
it has to be applied.
Yeah, okay. How it should be applied, right? Just for the second question, just you take the leaf, grind it in water, just apply it. Take the leaf, grind it by mixture, whatever it is, then spray it. That's all. No need of hot water or any other solvents. First thing, the 50% concentration. That means take 100 gram of this one and 200 ml of water. Okay, that will give you 50%. 1% means 1 gram in 100 ml. 1 gram in 1 liter, 0.1%. 1 gram in 100 ml, 1% or 10,000 ppm. This you just remember so that the calculation will be easier. 1 gram, 100 ml, 1% or 10,000 ppm. So 50% will be good. Your answer is for all your questions are answered. No, Jimmo, sulfur. Okay, sulfur you are asking. Yeah, the sulfur-based fungicides only we were using. Sulfur is fungicidal. It has got fungicidal effect. Sulfur, but other than that, there are so many other 16 different compounds we have isolated. In that, gallic acid, which is very good for heart attack. You can just browse it in Google. Gallic acid, you put it. It is very good for heart disease. So if you take even onion also, it is good for your heart. Okay, onion, both are same. Same means properties wise it is same. So okay, yeah. Thank you. Student side. Even from professor side, you can ask because it should be ultimately the students will be benefited if you ask some questions. Okay, from student side, 10 questions. Yeah, good, very good. No, don't fear for anything. Ask questions.
Morning,
See,
sir.
1 minute. In my class, whenever I attended here or somewhere, even in NAM after getting ARS, Agriculture Research Service exam, they will ask, that's all, the questions all is over or not. After asking that only you will finish up the lecture. So many people, everywhere. You have to ask, clear your doubts. For that only we are paid. We are here. Don't fear for asking questions, otherwise you will not improve. Anything, even if it is nonsense, no issues. Because you don't know, we don't know. For clearing the doubts, professors are there. You ask, keep on asking questions, then only you will improve. Yes, okay.
What are the basic nutrients required for the cultivation of banana, sir?
Basic nutrients required for
Cultivation of banana tree.
cultivation of banana. Okay. This generally primary, secondary and tertiary nutrients are there. Or macro and micronutrients are there. Earlier we were using only NPK, nitrogen, phosphorus and potassium. Nitrogen for the growth, phosphorus for root development and potassium for all increasing the yield, disease resistance, everything it is used. So they were using only NPK. And the FIM also, FIM contains everything, macro and micro. Now we are recommending micro also, because micronutrients is required in less quantity, but it has got so many functions. So you have to both apply macro as well as micronutrients. Micronutrients, like we have developed a micronutrient banana for banana as banana sakti, the micronutrients. Banana sakti, it contains copper, manganese, boron, and another one nutrients, iron. Boron, iron. Iron is a must. Manganese, magnesium, manganese, these are must. Copper. Boron also it is. Nowadays, because of over exploitation of soil, it is expressing the symptoms of micronutrients. That's why we are going for application of micronutrients. Okay?
Yes, sir. Thank you, sir.
How much it is required you want? NPK? Nutrients only asked, right? Okay. So I wrote one book in banana, A to Z, completely. It contains everything. In Tamil, in 2009 I have published, and for that I got Chief Minister Best Writer Award. So this banana book is there in most of the agriculture department in Tamil Nadu. The Tamil Nadu government has sent order to all the departments to purchase this book in Tamil. In English it is there, but afterwards I have not published in English version. But in Tamil it is available. Okay. First is over. Quickly. Good. Very good. See how people are asking very good questions. Organic farming.
For organically, apart from biocontrol agents, what are the management measures to control fusarium weed?
Apart from biocontrol.
Yeah.
Okay, good. Yeah, that's what I told you. The integrated management, the crop rotation, intercropping. Crop rotation is very, very important, because once the water is stagnated, paddy, suppose you take paddy, what we are doing, we are stagnating the water, right? Now drip irrigation and all it has come, but we are stagnating water. What our finding says, recently we found that 10 days if water stagnation is there in a field, the chlamydospores number is reduced much. So that type of things is there in paddy cultivation. That's why in the first year you will not get much incidents. Okay, paddy, intercropping, amendments, so many cakes and other things. This groundnut cake, all these things will improve the soil in the way of helping the microorganisms to grow, because it provides some nutrients for the microbes. Okay, so the beneficial ultimately everything whatever you do, microbes only, nothing more. And with regard to nutrients, the potash and the silica both are giving zinc. These three nutrients give very good resistance to the plants against disease. Okay.
Water stagnation is 10 days is possible for banana.
Water stagnation
For 10 days in banana is possible.
For 10 days is possible. Paddy if you grow, banana and paddy crop rotation if you follow, it is okay. Otherwise also there is a technique. Just to irrigate the land. Suppose maize star, paddy star, 500 kg you take, spread it in the field, irrigate it up to the field capacity. You know what is field capacity? Yeah, the water will be available in that, right? So for 20 days you maintain, it will reduce the enough level to a higher level. Suppose wherever the water is a problem. Okay?
Thank you, sir.
Online? Okay. Yeah. In the meantime, you can also ask questions.
What is the availability about this trichoderma and the Zimu plant for the commercialization of availability of the farmers?
Availability of?
Where these are available for the farmers? Where farmers can access this?
The biocontrol agents. You can get it from us. Very simple. Huh? No, that's what in my lecture I was insisting. Whatever trichoderma available with others may not work. May or may not work. It should be tested for 2 to 3 years under field condition, apart from testing it in vitro and pot culture condition. Because even in pot culture it will give 100% effect, but when it reaches to, when you put it in field, there are so many other factors are involved in that. You cannot, it is not in controlled condition, right? In the field, so many factors, nutrient deficiency, excess, then other microbes, other problem. With all this, irrigation may not be proper, or irrigation will be excess. In all these conditions, it has to survive, multiply, and act against the pathogen, right? So it should be any microbes developed or biocontrol developed, it should be tested at multi-location level for 2 to 3 years. Then only it would be effective for at least another 3 or 5 years. Otherwise, it will be a failure one. Okay. Jimmo, you are asking about Jimmo availability.
Yes.
I am very sorry because I thought that the biocontrol agents, you also agreed, but it is available in Periyakulam Horticulture Station and also in Kodaikanal, and with us also it is available. Okay. It is available. Very good. You can get it. You take it. You boil it and take it as a vegetable also. You can give it.
Sir,
For hostel people.
Another one doubt, sir. You said that if we treated the harvested fruit in that inoculum, it has a shelf life about 75 days. The quality will not be taken. What is the process, sir? If we control the disease and promote the growth in the field condition, that is okay, sir. But after harvest, the fruits shelf life will be how that is maintained. This ripening process and all.
So if you don't do pre-harvest spray, how it will control after harvest, right? That's what your question is. Pre-harvest spray you are asking, right?
No, sir. If நீங்க சொன்னீங்க சார் அதோட ட்ரீட் பண்ணா ஷெல்ஃப் லைஃப் வந்து 75 டேஸ் வரைக்கும் நல்ல ஃப்ரூட்ஸை வந்து கெடாம பாத்துக்கணும்னு சொல்றீங்களா சார்? இப்போ ஃபீல்ட் கண்டிஷன்ல அது வந்து டிசீஸை இன்ஹிபிட் அந்த ட்ரீட்மெண்ட் வந்து டிசீஸை இன்ஹிபிட் பண்ணுது அண்ட் ஆல்சோ growth-ஐ வந்து ப்ரமோட் பண்ணுதுன்னு சொல்றோம்ல சார். பட் இப்போ ஹார்வெஸ்ட் பண்ண ஃப்ரூட்ஸை நம்ம வந்து ட்ரீட் பண்ணும்போது அது எப்படி அந்த ஷெல்ஃப் லைஃப் எப்படி சார் இன்க்ரீஸ் ஆகுது அந்த ப்ராசஸ்?
Okay. The shelf life is due to two. This one is the pathogen will cause disease, and then by that way it will spoilage the fruit, right? Spoilage will occur. And another one, the ethylene it will emit. Okay, the deaminase enzymes, all these things you might have heard. This ethylene production can be controlled, should be controlled for increasing shelf life. That is the main factor. So this, by treating this one with GMO, and first the pathogen will be there as latent infection. You know latent infection. During green stage, it will be there in the fruit. Once if it starts ripening, then it will germinate and then it will infect this one fruit. So when you are doing pre-harvest spray, it will control the fruits. Okay. If you don't spray, harvest and you dip it in this one, it will, because latent infection, because it go into the system and kill the fungus present in that first thing, and it will prevent the release of ethylene in the fruit. When it ripens, ethylene will be emitted, right? So if you treat it with this GMO, it will not ripen for some time. So ethylene level is controlled and shelf life is increased. Not at room temperature, it is at 13.5 degrees centigrade. Okay, you have to keep it. Okay, the enzyme level will be decreased. Enzyme action will be not that much level at 13.5, right? And in addition to that, the GMO will be there. The principal compound will reduce the enzymes responsible for ethylene production. Okay, right.
Any questions from online? Okay, thank you very much.
Thank you for your nice presentation. I have one doubt. Told about diploid and diploid banana. So in Kanyakumari district, there is one diploid matti banana is available. So is there is any correlation between the diploid level and the quality?
Diploid level and
quality of the banana. That is nutritional quality.
No. Size is okay. Size will be size. There will be a difference in size because the diploid itself it is not that much in compared to diploid and tetraploid. So with regard to size, the diploid will be less because matti and naipun. This naipun is AB and matti is AA. These two are diploid. But the size, with regard to size, there is a difference. Not in other cases. Flavor and other things, it depends upon the environmental condition where it is grown. Suppose in cool climate, if it is grown, then it has got a very good flavor. Otherwise, it will not be having flavor.
Medicinal properties.
Medicinal properties, matti.
Yeah.
There is no much difference with regard to the constituents of alkaloids or nutrients in different varieties. We cannot say. There is no data on that. And that in general, the banana is having a lot of flavonoids and fructans. Fructans is the dietary fiber. So many things and vitamins and calcium. This is there per 100 gram. If you take any fruit, the level is almost the same. It's okay?
Yeah. In our laboratory, we have standardized the protocol for multi-microprocessor.
Okay. Have you noticed any difference?
Yeah.
In alkaloids level?
Not tested, but in our place they are provided to the small babies.
No, you compare with all other varieties, triploids and diploids, then you come out with some data.
Yeah. Thank you.
As far as what we have done, there is no much difference. But the sugar level, it is there. Suppose cooking banana, saba, then this one, Puvan. If you take the TSS content will be somewhat lesser than the other varieties like that probably, the Cavendish, TSS content.
You told that anthracnose infected fruit is very much useful for these cancer patients. So what is the quality of fruit when it attacks with the anthracnose pathogens?
Yeah, it's a good question. Yeah, quality will be reduced because though it is a skin deep, we will call it as a skin deep, but after some time it attacks the flesh also. If more severity is there, it attacks the flesh, and flesh will become rotten if the fruit is having more severity of the disease, and ripening will be faster. Spoilage will be more and ripening will be faster. So this is the effect of anthracnose. It has got effect on both the skin appearance also, because we will eat all these things, but not the other people other than India. Even if a small spot is there, they will not take it. They are afraid of taking this type of infected food. But before much ripened, there in foreign countries they take it, because after exporting our banana, then I went to Abu Dhabi and Dubai. I was watching the people how they are taking it, which type of food they are taking it, how they are consuming, to study all these things. The Lulu Mall, they invited us. We went there. So when we studied, before reaching to 5th or 6th stage, they are taking it. So they will not get, though the anthracnose, botulism, all these things are there in latent stage, they will take it immediately. So they are not facing the problem of the rotting due to the disease, since they are taking it at early stage of this one, ripening.
Thank you very much.
Thank you, sir. Now I request Dr. A. Soleimalai, Professor and Head, Department of Agronomy, to deliver the votes of thanks.
Respected and Honorable Vice Chancellor, Madam, Dean Agriculture College and Research Institute, Kiligulam, learned speaker Dr. Thangavelu Sir, Principal Scientist in CR, NRCP, Trichy, colleagues and student friends, good morning to all. It gives me immense pleasure in proposing the formal vote of thanks to the dignitaries and guests of this endowment lecture. On behalf of our college and Department of Agronomy, I first of all express my sincere, heartfelt thanks to our Honorable Vice Chancellor for presiding over the function. In spite of busy schedule, our Honorable Vice Chancellor, Madam, is very kind enough to participate and deliver the presidential address to the August gathering. Thank you very much, Madam. I also extend my sincere thanks to our Dean, SPGS TNAU Coimbatore, for offering welcome address and opening remarks to this endowment lecture. Thank you, sir, for permitting this lecture to our campus. Our sincere thanks to our respected Dean and beloved Dean, AC and RI Kiligulam, for giving welcoming address and also introduction of the speaker to the house. He is our guiding force for proposing, planning and meticulous conduct of the event in a grand manner in our campus. This is the first endowment lecture organized at our campus. Thank you very much, sir, for your guidance. Then I profusely thank Dr. Thangavelu sir, Principal Scientist, NRCP, Trichy, for nicely delivering the endowment lecture on current advances in plant disease management in banana. He is the pioneer scientist in the field of banana disease management. After hearing his lecture, he is very kind enough to accept our invitation to be the speaker of this endowment lecture. I thank our staff members of our college for organizing this program in a grand manner. I especially thank Dr. Joseph sir. He sent 2 times the proposal to our PG Dean. The third time only we got the approval of this endowment lecture, for which I thank our Professor Dr. Joseph sir. Finally, I thank the teaching faculties and students who have participated and graced the function. Thank you, one and all.
Thank you, sir. Now I request everyone to stand up for the national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मागे गाहे तव जय गाथा जनहन मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे